All right, kids, we're down to the final thing. If we look at the outline again, okay, we're down to the response. Okay, this Roman numeral 4 here. Okay, and we're really going to talk about cytoplasmic and nuclear. Fine tuning, we'll talk about amplification. Specificity, that's like the key in the lock thing. Efficiency and scaffolding, we'll look at a diagram on that. And termination is real easy. When that ligand or that signal detaches, then everything shuts down. Okay, so as long as that ligand is attached, that signal is attached to the receptor, the thing will keep going and going and going and going. It's like an on switch. But as soon as that is detached, that switches off. Okay, so let's talk about, though, the two types of responses, cytoplasmic and the good old dogma, the nuclear response. First, cytoplasmic. What I want to write here is this word, vesicles. Okay, we've heard of vesicles before with the formation of ER to Golgi to lysosomes. Let's write that down. ER to Golgi to lysosomes. So protein, okay, might be already packaged in that ER, but to have it get secreted out, it has to be processed. We know that the Golgi apparatus is where it gets packaged up and processed and transformed until it goes to maybe a lysosome, which eventually then gets secreted out. So vesicles are doing all that in between. They're jumping from here to there to there. But like I said, that may have been controlled by a nuclear response, but it doesn't get ejected until like another signal transduction pathway kind of triggers it to go out. When we look at epinephrine up close and impersonal, we'll see that epinephrine is the secretion of glucose out of the liver cells. It's a breakdown of glycogen into that glucose and gives you that sugar rush when you're on the adrenaline rush. Okay, that's a cytoplasmic response. Saliva in your mouth, I was talking about that one day. Okay, just thinking about something like bacon or a little Hershey Kiss or something. When you think of those things, it might make you drool a little, like Pavlovian dogs, right? That drool whenever they heard that bell ring. A dog will start to drool knowing that food's coming. And what it's doing is just like the thought of that food coming is actually triggering a signal transduction pathway, with the ultimate result being a cytoplasmic one where saliva starts getting secreted from your cells. Okay, so that's kind of crazy how that can happen. That's where an action can kind of like happen, or you actually eat the food and the saliva comes out. That's actually molecules then attaching to those receptors. But even the action, the nerve impulse or the hearing of that, talking about it, thinking about it, can actually like trigger that. But the dogma one is the more common one. These are the nuclear response. This is the DNA makes RNA, which makes protein. So let's see if you remember this. Should I do the awkward Dora the Explorer pause here? No. DNA, when it makes RNA, remember that was called transcription. Transcription. So a lot of times these responses, we call them transcription factors. So when a transcription, did I just move this out of the way there? Sorry about that. So when a transcription factor is actually the end result of that signal transduction pathway, that transcription factor will then attached to the DNA, which will transcribe RNA. And does anybody remember the next one? When RNA makes protein, that's also a TR word, but it's called translation this time. That's where like the tRNAs are matching up with the RNAs, codons to anticodons. It rings a bell, but it'll come back. That's like our next unit that we go into after this. Okay, we go into a lot more detail than you've ever gone in with this. Okay, and so there's our two responses. Okay, like I said, I'm just going to touch on these other things, okay, like amplification. I always like to describe amplification kind of like if I tell two people something and they tell two people and they tell two people and they tell two people. So amplification, I like to illustrate like one person tells two, but then they tell two. So it's like, and so forth and so on. And they tell to, and you can see that, through all of this, the response gets amplified. And this is very important with adrenaline, especially. Like if you think how fast you're scared and all of a sudden your heart's racing, that's how fast it takes to like release that glucose into your system. And a lot of it goes fast. It's like drinking 100 Red Bulls at a time. Maybe not that many, but close to it. So that's the amplification. Specificity. Always love saying that word. Specificity is like a certain shape fitting into a certain lock or a key in a lock. We know that ligands, must be specific to their receptors, just like substrates to their enzymes. And if you think of receptors, most of the time they are proteins. So it's got to be a substrate fitting into a receptor to trigger a response. That's what we mean by specificity. Then we have this efficiency and scaffolding. I want to show you another diagram that you have. And here it is here. Okay. And this is where I like to talk about like the variety of responses that can come about from just one signal. Okay. So let's look at this signal here. It's just triggering one response, one signal transduction pathway. But what if that signal comes in? It makes a molecule that splits into two molecules that would trigger two other responses. So we have this single response. Now we have two more responses that this little signal could have done. But what if that signal comes in and another signal comes in and it plays a whole role in producing this second compound here? That's a fourth type of response that we have. And then what if, and look at the difference here, what if it attaches to this protein, but it can also attach to this shaped receptor? So one receptor can accept that signal, but another receptor can accept that signal and produce a totally different response. Here's 5 different responses from like the same signal. So that's what I like to say when we talk about efficiency and scaffolding. Like that's where we're going to group signals and we're going to be more efficient because maybe it's not one receptor for every one pathway. Okay, maybe those G proteins and those ion things are a little more multitaskable, than we thought. Tyrosine kinases definitely can elicit like 6 responses. But imagine now with tyrosine, if we times it by 5 different responses that could even produce. Then we're up to like 30 responses that one type of receptor can handle. And finally, so that was like, and I'm looking at the outline here. So just so you can see, I'm looking at these fine tuning the response. Finally is termination. I'll write that down right here. Okay. Termination is simply, when does this all stop? Like when do people go to bed? Okay. Termination. Okay. Termination. We know when a signal, and I'll just draw it kind of like this, is attached in there, it's going. Okay, so when there's a signal, let me label this signal, and it's attached to the receptor, okay, then it's on. It's like the on switch, okay, it's on. But once that signal detaches, so if this is the signal, right, and it detaches from this thing, here's a little attachment area, right, once it detaches, then it's off. Okay, and that's how you shut down these signal transduction pathways. Sometimes it will be a feedback thing, a negative feedback thing, where the end result will pull that away. That's where we talked about that in the last unit. Okay, and most of the time it will be that actually. So going is when it's attached, off is when it detaches. And that's it for this video.